Hello everyone. Now we are coming to the end of karyokinesis, and last stage of karyokinesis is telophase. We have already discussed in detail about the different changes that occur in the cell during prophase, metaphase and anaphase. Now the telophase is the last stage in which one nuclei finally get converted into two daughter nuclei. Okay, and now the formation of two daughter nuclei will going to take place at two different pole, and this will be the end of karyokinesis. And now cell is ready. After telophase, cell is ready to enter in the cytokinesis. In this video, I am going to discuss in detail about the different changes that occur in the cell during the telophase. As I discussed in my last video, telo means end and phase means stage. This is the stage which end the process of karyokinesis. Okay, before starting with the telophase, I just want to give quick recap of the anaphase. In anaphase, sister chromatid split from one another, okay, due to splitting of the centromere, and now due to contraction of the spindle fiber and pushing action of the interzonal fiber, now this daughter chromosome will migrate towards their respective poles. Okay, so at the start of the telophase, you can see something like this: the centriole at both the end. Centriole have aster rays, short fibers, and they also have long fibers called spindle fibers. And now due to the contraction of the spindle fiber, these chromosomes are already reached to the poles. We have take example of cell which having 5 chromosomes. So now this pole will going to have 5 daughter chromosome, and this pole will going to have 5 daughter chromosome. Once the telophase start, the cell will going to perform the activities exactly opposite to that of the prophase. So if you want to remember telophase, okay, just understand the prophase. Okay, all the activities which is happen in the prophase will going to get reverse in the telophase. So first thing is that we have written that in early prophase, nuclear membrane start disappearing or start dissociation of the nuclear membrane. So in this case, first thing happen is nuclear membrane reappearance. Okay, assembly of the nuclear membrane and reappearance of the nuclear membrane will going to take place at both the pole around the daughter chromosome in telophase. So this is the very important change that occur in the cell during the telophase. Next thing happen is that condensation of chromatin if we consider prophase. So here decondensation of the chromosome. Okay, in prophase, due to condensation or removal of the water molecule, chromatin get converted into chromosome. But in this case, due to recondensation, water molecule come in the picture, and due to condensation of the chromosome get converted into thin, long, thread-like structure called chromatin. Okay, this is another important reverse process which occur in the telophase. In prophase, conversion of the chromatin into chromosome due to condensation, while in case of telophase, conversion of the chromosome into chromatin due to recondensation. Okay, so the next point is formation of chromatin, obviously from chromosome, by recondensation. So now due to nuclear membrane reappearance and formation of chromatin from chromosome by recondensation, now both the pole will going to look like this. Instead of chromatin, now you can see 5 thread-like structures, which is chromatin at both the pole. Okay, when we compare structure of chromosome, it is thick and short with chromatin which is thin and long. Okay, you can clearly find the difference, and this is happened due to recondensation process. At the same time, nuclear membrane start reappearing. So here you can see a nuclear membrane start reappearing around each set of the chromosome or chromatid at both the pole. Third change that occur during the telophase is centriole migration. Okay, in this case, centriole migrate towards opposite pole. Okay, and they become active and they start synthesizing spindle fiber. Now, in this case, centriole disappear. Centriole disappear, and this spindle fibers fiber dissociate from the chromatin and disappear. Spindle fiber disappear. Okay, so third important point is that disappearance of the centriole as well as dissociation and disappearance of the thread, that is spindle fiber. And due to this, now both the pole of the cell only have nuclear membrane, and inside the nuclear membrane you can clearly see one set of chromatin. Okay, another change which take place in the telophase is uh, migration, disappearance. Okay, we know that a nucleolus which is present in the nucleus also disappear. Nucleolus also disappear. So in this case, a sessile body which is present in the nucleus, that is nucleolus, reappear. So another important thing is that reappearance of nucleolus. So I can show one nucleolus in this nucleus and another nucleolus is in the next nucleus. Okay, so if we consider telophase and prophase, the telophase is exact reverse of the prophase. All the changes that occur in the cell during the prophase get reversed during the telophase, and at the end of the telophase, now we will going to have cell with two daughter nucleus. Okay, nuclear membrane assembly completed, and cell will going to show presence of two daughter nuclei at both the end. Okay, with nucleolus. Spindle fiber disappear, centriole disappear. Okay, and chromosome get converted into chromatid due to recondensation process. So this is all about the telophase. At the end of telophase, you can going to get one cell with two daughter nuclei, and now this cell is ready to enter in the last stage of mitosis, that is cytokinesis. In my next video, cyto means cytoplasm and kinesis is division. In my next video, I am going to discuss in detail about the process of cytokinesis, in which now due to division of the cytoplasm, one cell will going to form two daughter cells finally, and that will be the mark of end of mitosis. Thank you.